Good morning, my dear children. I'm very happy to meet you all back in the biology class in the term 2. The beginning is the most important part of the work. No matter how hard the past is, you can always begin again. Begin always. So as we're beginning this new term 2, I pray to God that God will enable each and every one of you to do well in your academics and other activities too. So what is God expecting us to do at the beginning of this term too? As it says in Ephesians 5:1 and 2, therefore be imitators of God as beloved children and walk in love as Christ loved us and gave himself up for us, a fragrant offering and sacrifice to God. With that godly thought, with a Christ-like thought, let's go into today's lesson. Today we're going to start with Biobotany, lesson number 5, plant tissue culture. So what is plant tissue culture? The growing plant part, which is nothing but the protoplast, the cells, the tissue, the organ under natural or artificial condition. It is also called in vitro growth, that is growth outside the body in the laboratory. The single plant multiplied into thousands of plants in a short span. The scientists called Gottlieb Herbert Land in the year 1902 proposed the concept of totipotency and is also called the father of tissue culture. Let's learn more scientists who involved in the study of plant tissue culture, the milestones in the plant tissue culture. The Herbert Land cultured plant cells artificially, that is in vitro. Later, the scientists called Guha developed the in vitro production of haploid embryo. The scientists Canty et al. In the year 1962 produced test tube fertilization in the flowering plants. Tegbe et al. He produced regenerated plants from the tobacco. P.R. White developed the root culture with vitamins. Melchers developed the hybrid potato plus tomato. And Carter et al. He produced test tube fertilization with the flowering plants. Now, these are some of the scientists involved in the study of plant tissue culture. Let's learn the basic concept of plant tissue culture. What are the basic concepts? The 4 concepts are totipotency, differentiation, redifferentiation and dedifferentiation. Let's learn the basic concepts one by one. The first one is totipotency. Now what is totipotency? It's a property of a living plant cells with genetic potential. When cultured, it gives rise to new complete individual plant. The second one is differentiation. Here, the biochemical and structural changes occur. The cell becomes specialized in form and function. The third concept is redifferentiation. Here, the further differentiation of already differentiated cells into another type of cell, the callus to a whole plant in a nutrient medium. The last one is dedifferentiation, the last concept. Here, the reversal of mature cells into bedsomatic cells. It forms callus. It inherits a property of living cells. Let's learn more about PTC, plant tissue culture. What is plant tissue culture? It's an in vitro culture, that is culture outside the plant, that is in the laboratory, in the aseptic growth of any plant in a tissue on a tissue culture medium. So what are steps involved in the plant tissue culture? Step 1. Here the plant part must be selected and isolated from a plant body. Step 2. The explant must be controlled physically and chemically, which is under defined condition. The third step is the explant, the product. The tissue is taken from the explant, nothing but the tissue taken from the selected plant, which is transferred to a cultured medium, form a new plant. You can see this diagram. The basic steps involved in plants tissue culture. Here you can find a part of the plant removed from the tree, any part, the leaf or stem or a root. Here it is cultured in the aseptic medium, the culture medium. Later it grow into a callus. This part of cell from the callus is removed. They undergo the callus separated and undergo culture, single cell culture. Later the culturing produce new plant, regenerated new plants. So how will the lab perform? What is the main part of the lab here, laboratory here? Here the lab play a very major role, the scientific laboratory. The washing facility for glassware and ovens for drying glassware. All this should be available in the laboratory. The medium preparation room with autoclave, electronic balance and pH meter. The transfer area of sterile room with laminar airflow bench. Apart from this, the culture facility for growth of explant also occurring inside the lab. This lab play a very major role in the plant tissue culture. Let's see the techniques of plant tissue culture. What are techniques involved in plant tissue culture? First one is sterilization, induction of Callus, embryogenesis, media preparation, hardening, finally the culture conditions. Under sterilization, what is sterilization? It's a technique employed to get rid of all the microorganism, virus or bacteria in the culture medium, the vessels and even from the explants. So what are the steps involved in the sterilization? Here the maintenance of its aseptic environment. The sterilization room, a culture room play a very major role here. The sterilization of nutrient media and the sterilization of explants. All these occur under the sterilization condition. During maintenance of aseptic environment, the sterilization of glassware, forceps, scalpels and all accessories in wet steam by autoclaving at 15 degrees Celsius for 15-30 minutes or dipping in 70% ethanol followed by flaming and cooling. Sterilization of culture room. Floor and walls are washed first with detergent and then with 2% sodium hypochlorite or 95% ethanol. The cabinet of laminar airflow is sterilized by clearing the work surface with 95% ethanol and then exposure of UV radiation for 15 minutes. Here the culture media are dispensed in the glass containers plugged with non-absorbent cotton or sealed with plastic closure. An autoclave at 15 degrees Celsius or 121 degrees Celsius for 15 to 30 minutes. The plant extract vitamins, amino acid, hormones are sterilized by passing through millipore filter. Sterilization of explants. The plant materials to be used for tissue culture should be surface sterilized by first exposing the material in running tap water and then treating in the surface of sterilization agents like 0.1% mercury chloride or 70% ethanol under aseptic condition inside the laminar airflow chamber. Second technique is media preparation. So how are the media is prepared? Now what is media? It's a place where you culture or grow the explant of the plant. The suitable medium as per the principle of tissue culture is used. Whatever medium we need for the particular plant will be produced. The commonly used medium are AMS medium, which is a carbon source with vitamins and number of hormones for the plant's growth. Apart from this, there are some other mediums like B5 medium, white medium, niche medium, etc. So the medium may be solid, semi-solid or liquid. Agar is used for the solidification of the medium. The third technique is culture condition. So while plant breeding, there should be a perfect culture condition for the plant tissue culture. First one is the pH, the amount of hydrogen atoms, normally adjusted between 5.6 to 6. Temperature. Culture should be incubated at constant temperature about 25 plus or minus for the optimal growth. Humidity. The culture requires 50 to 60% of relative humidity. Light also play a very major role in plant tissue culture. 16 hours of photoperiod by illumination of cool white fluorescent tubes. Aeration, presence of air play a very major role here. It provides by shaking the flask or tubes of liquid culture by passing the filter sterilization air. The fourth technique is induction of Callus. The explant of 1 to 2 centimeter from the sterile segment is selected from a leaf, stem, tuber or a root. Secondly, it is inoculated in the medium. Here is MSPDM with auxins. Auxins are hormones which help the plant's growth, and incubated at 25 degrees centigrade. It alternates the light and dark period of 12 hours to induce cell division and develop the culture. Finally, the callus, a mass of unorganized growth of plant cells, are produced in the in vitro culture medium. The fifth technique is embryogenesis. Here, the callus cells undergo differentiation which produce somatic embryos. The embryoids culture again to produce the plantlets, small plants inside the test tubes like this. Finally, hardening. What happens in hardening? Gradually, the exposure of the in vitro development from the test tube, it's passed to next stage, where the humid chambers and diffuse light, which enable them to grow in the hardened surface, maybe the normal field conditions also. Let's look at the types of plant tissue culture. There are 4 types: organ culture, meristem culture, protoplast culture and cell culture. Under organ culture, the culture of embryo, anthers, ovaries, root, shoot on the culture media occurs. In the meristem culture, the culture of any plant meristematic tissue on the culture media. What is meristematic tissues? The young tissues. Under protoplast culture, there are 4 steps involved: the selection of somatic hybrid cells, culture of protoplast, fusion of protoplast, and finally isolation, separation of the protoplast also occur here. Under cell suspension culture, the growth of cells in vitro liquid medium. The cell suspension is prepared by transferring portion of liquid medium and agitated, mixed thoroughly. So what are the product of secondary metabolites? The secondary metabolites are produced at the end of the plant tissue culture. Some of the metabolites are digoxin, which is a plant source of digitalis purpurea, the use of it cardiac tonic. Next one is codeine. It's a plant source of Papyrus somniferum, used for analgesic. Next one is capsaicin. It's a plant source of Capsicum annum, used in the rheumatic pain treatment. Next, vincristine, Catranthus roseus, anti-carcinogenic. Finally, quinine, Cinchona officinalis, used as for malaria, anti-malarial treatment. Okay, these are some of the secondary metabolites we have learned in this lesson. Now finally, the recap what we have discussed in today's class. We have started with the introduction of plant tissue culture. Then we discussed about the milestone of plant tissue culture. We learned about number of scientists discussed or discovered about plant tissue culture, techniques involved in plant tissue culture, types of plant tissue culture, and the product of secondary metabolites. Children, we have completed the first topic of today's lesson, that is plant tissue culture. Go through the video carefully, read the lesson from the textbook, understand the meaning of it, then watch the video, prepare for the test as well as Kahoot or quizzes. Make sure you attend the test, Kahoot or quizzes regularly. Prepare well for your board exam. That's all from today's class. Have a nice day and goodbye.